So, a very good morning to all of you. Thank you very much for joining us in time. And we are about to start with the fourth day of this short-term training program on biotechnology and its application in tribal agriculture areas, which is sponsored by All India Council for Technical Education and organized by Ballarpur Institute of Technology, Ballarpur. Our honorable resource person for today's session is Dr. Harit sir. However, before we begin with the session or before we move towards starting the session, first, I would like to take this opportunity to wish all of you a Merry Christmas. So, Merry Christmas to all of you, and may the new year bring joy to all of us after this. We have already seen that the year 2020 has been really very challenging for all of us. Let's hope that the coming year after this Christmas brings some good news and great news, and we are all in good health. So wish you all a lot of health, happiness, and prosperity on this eve of Merry Christmas, and also to our honorable resource person, Dr. Harit sir. Wish you a Merry Christmas, sir. So it is time to introduce our honorable resource person. Dr. Harit sir is professor in Department of Biotechnology of Guru Ghasidas Vishwavidyalaya, Bilaspur. He has completed a PhD in biochemistry from Rashtrasant Tukadoji Maharaj Nagpur University. He has qualified SET lecturership from Maharashtra in February 2002 and CSIR UGC NET lecturership in June 2002. He has published more than 30 papers in national and international journals. He has also jointly authored two books. They are named as Antioxidants, Source Activity and Applications, and Lignocellulose Based Composites and Biopolymers. Several projects that have been sanctioned and completed under his leadership include CCOST funded project worth rupees 5 lakhs on lignocellulic polylactic acid based composites for multifaceted applications, a 3-year project on application of acacia and rice husk lignin in the synthesis of biopolymers, biofilms and resins worth rupees 11 lakhs and 26,000. He was a group leader in the 5 years project on microbial technology under DBT Builder Scheme to boost interdisciplinary education and research in School of Life Sciences, Biotechnology, Botany, Zoology. This was again of DBT. This project was worth rupees 3 crore 69 lakhs and 80,000. He has successfully completed a 3-year UGC funded project worth rupees 10 lakhs 4,000 on process optimization of conditions for extraction and purification of lignin from agro and forest wastes using physico-chemical and biological approaches. Furthermore, he was deputy coordinator in a UGC SAP2 DRS1, a project worth rupees 87 lakhs 50,000 for Department of Biotechnology of Guru Ghasidas Vishwavidyalaya. Dr. Jha is also a PhD guide. As on date, 3 PhDs have been awarded under his guidance. His expertise has earned him to be a reviewer for international journals like Material Science and Engineering, International Journal of Biological Macromolecules Chemosphere, Romanian Journal of Biotechnology, Pharmaceutical Biology, and Journal of Nanoparticle Research and Bioresources, etc. Well, I must say, Dr. Harijha sir, we are really glad to have you as our resource person today. And I would also like to share this information that Dr. Harijha sir has been the recipient of Best Speaker Award for our previous short-term training program. So I suppose that all of you, you will enjoy his session a lot. And may I now request you to please start the session, sir.
Thank you, Abhijit. And now I'll be starting with the presentation because we are already a bit late. Merry Christmas to all of you. Uh, as I already mentioned that I'll be presenting on agro waste and especially the rice and babool. Now, how exactly we can use the resources which are available with us? That is what is my concern. Now, my presentation is visible to you.
Yes, sir, it is visible.
Okay. So my topic is bio-based products from agroforestry waste, and we'll be also taking example of rice and babool. Now, India actually generates over 400 million tons of agroforestry residues, This agricultural and forest biomass are available in large enough quantity to be considered for large scale production of alcohol based fuels for many other applications. Now, the worldwide potential availability of this non-woody material is estimated to be 2.5 billion metric tons per annum. One of this, that is, rice, is an annual crop, and one of the byproducts is rice stalk. Rice stalk can also be used as source of biomass and lignin. The agro waste that is coming from the agricultural operations produce large amount of byproducts that have generally very low economic value, like rice straw, wheat straw, husk, sorry, stumps, barley straw, grass seed straw, flax coil seed, corn stalk, sabai grass, cotton staples, sugarcane, bagasse, on which we have also worked during my PhD. Some of the waste is utilized as fertilizer, fodder, or as fuel. A large utilized. So a strategy is required to convert this so-called waste into value-added product. The process of this conversion should be technologically efficient, economical, and with ease of operation, so that a common farmer should be able to optimally utilize its resources to generate secondary source of income. So what we have seen in agriculture sector is that, and especially even the agroforestry industries, that uh, or including the farmer, go for the main product. That is, for example, if it is grain producer, so they will go for rice grain, or wheat grain, or forest producers are there. So they will use, for example, timber, and the remaining byproducts are normally low cost, and they are mostly used as fuel of or as feed or fodder. So we are not utilizing them efficiently. If we utilize those forest or agro producers efficiently, and every part of them is converted into value added product. Value, when we talk of value addition, it means that the cost of that particular product should increase. So whatever byproduct which we are treating as waste should be converted into something which is useful, viable, and have higher cost. So that can be done. If we do that, then we add to the income of the farmers. We add to the income of the tribals who are collecting the forest produce. So basically, the sources of biomass or carbon-based substances are from forestry, agriculture, residue, sewage, municipal solid waste, industrial residue, etc. And the traditional use of this forest and agro waste is as fodder, as fuel, as timber, and mainly the non-agricultural products and produces are using fuel. So when we burn something, that's fine, because sometimes it is essential as fuel. But when you burn something, it is like firecracker. It gives a lot of bang and sound for a few seconds, but after that, that 500 rupees or 1,000 rupees is gone. So when we start treating this waste as the money, as the value added product, then only we'll be able to increase the overall economic situation of the farmer. Farmers are under distress, there is no doubt on that. But one of the major reason is that they have to rely on only one product which comes out from their field. If all those which is coming out from field can be converted to useful and costly products, obviously they can gain a lot. Now, taking an example of rice, in this presentation, first I'll be discussing about rice and babool, and how each and every part of their whole plant can be utilized. And then we'll look into the individual cellular components or biochemical components of the plant or agro waste. And from individual those biochemical components, how we can enrich ourselves, how the useful products can be obtained. So we'll take an example of rice and then example of baboon, and then we will move into the individual components of agro waste, and then how exactly the individual components can be utilized efficiently. I'll be also discussing some of the work which we have actually done. So rice or rhiza sativa has three major components in the plant, that is stalk or straw. Okay, then rice husk and bran, and rice grain. So this is rice husk, this is straw, and the whole plant. A rice grain is a staple food, and besides being used as food, if it is fortified, it may provide sufficient nutrition or nourishment for the undernourished population. So we have seen the example of golden rice in which vitamin A was added or fortified. We have also seen the example of in India, especially in the Indira Gandhi Agriculture University, they are working on the iron fortified rice grain. So that iron fortified grain can also help in improving the nutrient value of the rice grain. So rice grain is a staple diet, and obviously it is the main crop. It is the main product from cultivation of the rice. The cost may vary from 1600 per quintal to 2500, which is being provided in one of the states. And if the grain length is long and the quality of the grain is good enough, in that case, they can get higher prices. But the price may vary between that 1600 per quintal to say 2500 to up to 2000 if it is basmati, and 3000 if it is a basmati variety. Now, one of the major issues with the rotten grain, that is the FICCI, the FCI and other people, they purchase the grain, but that is damaged due to pest, weather, moisture, or transportation. Uh, but that can be used for the fermentative production of ethanol, butanol, methanol, glycerol, single cell protein, lysine, and other major solvents and therapeutic commercial products. So even if the rice grain is rotten or damaged, it can also be used as a source of carbon for the fermentation, for the fermentative production of the various different types of solvents and chemicals. So it's not that when we read a lot about rotting of grains in large godowns, uh, we feel that it has lost its value. No, it has not. If we can find a proper way by which even if it is rotten or moist, that moisten or rotten grain can also be used for production of the useful chemical. In that case, obviously, it will help in value addition. Then coming to the rice husk and bran, they are the major source of rice bran oil that is rich in many oxidant and antioxidants and therapeutic agents like omega-3 and gamma oryzanol. So obviously many of we people are purchasing the rice bran oil, and it is quite costly also, and it is said to be good for heart. So that is again a value addition where another so-called waste product coming from the rice is being used for production of a very useful or value-added product. This rice husk, even after extraction of the oil, is also a source of cellulose, hemicellulose, and lignin can be used for various different products. Third component, which is rice stock, can be treated to make them suitable for major products. For example, alcohol. Fuel grade cellulosic ethanol can be produced using pre-treated stock to make it permeable to microorganism and microbial fermentation. Rice straw is also a rich source of cellulose and hemicellulose, and this straw can be treated again with alkali to isolate cellulose that may find use in making paper and packaging material. The cellulose derived from rice straw can also be used for making clothes. So, because cotton clothes obviously contain cellulose fibers coming from the cotton. So, if we can uh, uh, extract the cellulose long length without without much breakage, the rice straw cellulose that can be also woven and can be used for making of the cloth. Rice straw can be combined with lignin and other plasticizers and cement to make composites. Rice straw is also source of lignin and silica that are byproduct after extraction of cellulose. This lignin can be then used as standing agent, dye, fuel, binder, resin, etc. for making plywood as cementing material, and many other uses are there for the lignin. The silica which remains after extraction of lignin and cellulose can be used as component of cement and composites, and that imparts strength to the building material. These are some of the examples. Normally, what we do is that you have to burn. That burning can also create many problems, including uh, the pollution, the air pollution. But instead, if you can use this particular straw for converting into fibrous material and then polymers and plates, uh, biodegradable material. These are some the example. This is the rice bran oil, which can be obtained from rice bran and rice husk. These are the composite material which are being used, or hardboard, which can be used for making the furniture. This can be used for food plates and other materials. These are packaging material which can be made from the rice straw. So instead of just wasting that particular straw, if we can purchase it and convert it into these useful products, no doubt the value addition will be there and the farmer's income will grow. Right now, we don't have any system where this farmers will be able to utilize those and convert them into useful products. But yes, at say village level, if we can find a small scale unit where whatever rice straw, husk, and other components are present, they can be converted into a useful product. In that case, that will help a lot to the farmer because they will be able to sell their not only main product, that is rice grain, but other byproduct to a use to a good amount, and that will help their economic condition. Another example which I have taken is babool or acacia. So there are more than 1,000 species of acacia. One of the prominent acacia species is Acacia arabica and Acacia nilotica in India. Now, this acacia or babool is also considered as unwanted plant, and in mostly the arid and semi-arid region, this babool can grow quite easily. Uh, the major uses of this is the distribution of babool in most of the arid and semi-arid region. This babool is present. This is structure of the various parts of the babool, including flower, leaf, bark, and fruits, etc., which are quite useful. Now, what are the major uses normally of this acacia species, which is known as babool? It's no medicine. They have many long been used for the treatment of skin, sexual, stomach, and tooth problems. In addition to utilizing this edible seed and gum, which is called acacia gum, we also get a sticky uh, polymeric gum-like material from the acacia, which is called the acacia gum, which is quite costly also. In addition to the utilizing the edible seed and gum, the people employ the timber for implements, weapons, fuel, and musical instruments. Uh, so you can see this is also a comparison between timber and the acacia or babool wood. It's quite useful, and especially for the relatively cheap furniture, the babool is quite useful. This is one example of babool fali, that is pots, babool or acacia pots, which are used for reducing the joint pain. These are some of the other products. Here you can see that there are 90 capsules of babool fali which is being sold. Okay, this is babool gum or acacia gum, which we also use in food and other materials. These are again fully. Then is the bark and its powder, which is being used as in ethnomedicine, especially the Ayurveda. Then this is Babool, obviously well-known brand also, which claim that the Babool powder is present in that, and it helps in removing the tooth-related ailments. So these are two examples which I was saying that whether it is a unwanted plant like Babool, or it is the rice which we are talking, or rice husk and rice straw, which are unwanted. Now, if we can convert them into useful components, then obviously the value addition will take place and the condition of the farmer will improve a lot. The only thing is to think about starting the smaller units where we can convert a tinkering lab, rather, we can say, where we can convert in each village in each taluka the these waste product into useful product, and if we can have a sale of those useful product in uh, uh, specific outlets or melas, in that case also it will help in uh, improving the publicity, sale of those particular alternate products, and also will help in the overall village economy. Now coming to a bit more scientific part of the talk. Uh, when we talk of the composition of agro waste or even the forest waste, normally contains cellulose, hemicellulose, lignin, and other components. So it is also called as lignin cellulose or lignin carbohydrate complex. So of this, cellulose and lignin are the most abundant material on the earth because they cannot be easily degraded and they keep on accumulating. Although there are a consortium of microorganisms which are present, including many basidium such as white rust fungi, soft red fungi, brown red fungi, which can degrade or partially transform the lignin and cellulose, but it takes time. So comparing it to the large bulk which is present, it takes time to degrade whole of the cellulose and lignin, a whole log. If you want to degrade the whole, a complete log of a tree, it takes years for its complete degradation. So hundreds of years will take place for complete degradation of that particular log if you leave it in natural condition. So that is why they keep on accumulating. But they can be these cellulose and lignin can be then utilized for the production of value-added products such as biodegradable film, foam, resins, coating, for drug delivery material, for nanocomposites, for nanomaterials. Now, these materials, the lignin and cellulose and hemicellulose, although they are bioaccumulating in nature, their large bulk is present. And best part is that it is renewable in nature, that every year you get a large cache of this cellulose lignin. So you can keep on utilizing them for many useful products. Now, this lignocellulose refers to plant biomass, which is composed of cellulose, hemicellulose, and lignin. And they are this carbohydrate polymers like cellulose and hemicellulose are tightly bound to the lignin. Uh, these lignocellulosic biomass mainly come from agricultural residues, dedicated energy crops like jatofa, karanj, babool, etc. Then wood residues, including that of sawmill and paper mill discards, then municipal waste paper. So all this material are the major sources of lignocellulosic biomass, which can be utilized for converting them into useful products. Now, this lignin carbohydrate complex is generated during the conditions of lignin synthesis, biosynthesis, which is a free radical mediated process during the nucleophile supplement to phenol methide. The intermediate connections are developed due to para-hydroxycinnamyl alcohol oxidation process. These lignin carbohydrate complex linkage play a crucial role in wood structure since all lignin molecules in softwood and 47 to 66% of lignin fragments in the hardwoods are bound to carbohydrates, mainly to the hemicellulose. There are 8 different types of lignin carbohydrate bonds, including benzyl ether, benzyl ester, glycosidic or phenyl glycosidic, hemiacetal or acetal linkages, and ferulate or diferulate esters that are linked to lignin using 4 hydroxy and for orthopositions. So these are the glimpse of some of the bonds with carbohydrate lignin and carbohydrate linkages. 8 different type of linkages which can occur between lignin and hemicelluloses and lignin and cellulose. So cellulose is an organic, another component of this lignin cellulose complex is cellulose. It's a major component. It is a polysaccharide with a linear chain of several hundred to 1,000 glucose units. And by Kraft process, normally we separate the cellulose from lignin in the plant, uh, from the plant matter in the paper mill. This cellulose then becomes the major constituent of paper, paperboard, cardstock, textile, including the cotton, linen, and other plant fibers. This is the overall structure of the lignin. Crystalline cellulose material, the microfibril structure, and its location in the plant cell wall. The second component of lignin carbohydrate complex, which is present in the plant biomass, is hemicellulose. Uh, Hemicellulose, it's basically a heteropolymer. It contains many different type of components, including xylene, gluconoxylene, arabinoxylene, glucomannan, and xylomannan. So these polysaccharides contain many different sugar monomers, whereas in cellulose only anhydrous glucose is present. Many of the monomers include glucose, xylose, mannose, galactose, rhamnose, and arabinose. Mostly the hemicelluloses contain D-pentose sugar as well as some of the L sugars, although the L sugars are in small amount. And cellulose is a major monomer which is present along with the mannuronic acid and galactonic acid. Then coming to the third part, lignin, and this is the molecule which interests me most. And this is the molecule uh, which helped me to get my PhD and PhD of 3 more students, as well as it also helped me uh, to get interested in the field of research. Now, this lignin is the most abundant polymeric carbon after cellulose. It's a three-dimensional network molecule, which is built up of dimethoxylated and monomethoxylated and non-methoxylated phenylpropanoid units, which are derived from corresponding para-hydroxycinnamyl alcohol. These units combine together with different types of bonds, including ether and carbon-carbon covalent bonds, to form a polymeric structure of lignin. This lignin normally is considered as waste, and as we have already seen that they are bottleneck to the overall carbon cycle, but they can be converted into value-added products, including biofuels, cheap energy sources for fermentation, and they can be broken down so that they can improve the animal feed and animal nutrients. Now, these lignocellulosic enzymes can be used for this lignin breakdown and conversion into the smaller monomers as well as useful products. Now, various different type of value-added chemicals and products can be obtained from lignin. Example, syngas products like methyl alcohol, dimethyl ether, and mixed liquid fuels. Hydrocarbons such as cyclohexane and higher alkylinates, then phenolic compounds, oxidized products like dimethyl sulfoxide, then aldehydes, quinones, vanillin, vanillic acid, and other aromatic and aliphatic acids. Macromolecules, for example, activated carbon, carbon fibers, adhesives, resins, polymer alloys, polyelectrolytes, nutraceuticals and drugs, then wood preservatives, substituted lignins, thermosets, and composite materials. Moreover, lignins are also used in foams, dispersants, emulsifiers, and emulsion stabilizers, binders, sorbents, wood like thermoplastics, then ion exchange resins, water treatment for they help in softening of the hard water, then as pesticide, as sequestering agent, animal feed, grinding aids, and concrete additives. So sometimes when someone hears that there are so many applications, it raises question that is it so useful that it can be used almost everywhere, or we are just attributing many properties on uses to the lignin just because we are interested in on working in lignin. So unless you see exactly what happened with lignin, unless we see that yes, it can be converted into useful product and quite efficiently, we cannot uh, be sure. We cannot be uh, 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 confirm that it can it can be useful. So I'll be also discussing some of the publications which we have, some of the work which we have done with the lignin, where we have used and converted the lignin to many different useful products. The profitable utilization of lignin can be possible by various chemical and biological modification. These include alkylation or arylation, carboxylation, polymeric condensation, products with phenols, aldehydes, and amines. Then phenol derivatives with salt of heavy metals, nitration, halogenation, including chlorination and chlorobromination, or oxidation. Other reactions include oxyalkylation, demethylation, sulfonation, or sulfomethylation to produce lignin sulfonate, and many other unit processes. Some of the processes are established in industry, but the majority of these are yet to be developed in commercial scale. Now, this is a major flaw, like including, I'll include myself in that also, that most of the processes which are developed either in industry or during the research work, they are not upscaled, like they have not been executed at the large scale and commercial setting. So unless we do that, their market will not increase, their productivity will not increase, and their application use which we intend cannot be obtained. Now, other products which can be obtained from the lignin include, obviously, it is a polyphenol, so lignin can be broken down or modified uh, to produce many different phenolic compounds. So normally they are derived from the petroleum industry, but lignin can serve as an alternative source for production of various phenolic compounds. Lignin and phenolic compound derived from it can substitute petroleum derived phenol for various industrial applications such as adhesive, resins, and surfactants. Now, the potential application of lignin derived phenolic compounds are in agriculture sector for making herbicides, in leather industry for tanning of animal hides, food, cosmetic, and pharmaceutical industry. Then vanillin, vanillic acid, cresols, coniferols, eugenol, and syringol are some of the important phenols which are derived from lignin. Another lignin-derived phenol compound is glycol, which acts as a precursor to other phenols such as eugenol and vanillin. Glycol and its derivatives are also used in pharmaceutical industries as expectorants, as antiseptic and local anesthetics. Vanilla is also a natural flavoring agent which is mainly extracted from beans of vanilla plant like Vanilla plantifolia. Uh, Vanillin and vanillic acid also can be derived from the lignin and can be used as an alternative to vanilla extract. So they will be a lot more cheaper as compared to that which is being obtained from the vanilla plant. So these compounds then can be used in the food industry as flavoring agent or in cosmetic industry for fragrance and in pharmaceutical industry. Another application is of lignin-based composites, of lignin-based composites. So softwood and hardwoods are good source of this lignocellulosic fiber. And for example, different plants, part of the plant like leaf, bark, seed, and fruit can act as a major source of the lignocellulosic fibers. Then fibers can be also obtained, bark fibers can be also obtained from inner bark of flax, jute, hemp, mesta, and Iyakawa plants. Cotton and kapok are also good source of seed fibers, while coconut and loofah are good source of fruit fibers. Grass and reed fibers are another class of lignocellulose fibers can be obtained from wheat, rice, and oat straw, bamboo, sugarcane, bagasse, elephant grass, etc. These wood and non-wood lignocellulose fibers are used for making papers in pulp industry and for manufacturing biocomposites. These biocomposites can be used in making automotive components, non-food packaging, and biomedical engineering for repair and restoration of tissues and implants, and for delivery of drugs and genes. Lignins can also be combined with polymers to increase the mechanical and other physical property of the combined material. And being an antioxidant agent, lignin can be used as stabilizer to prevent or reduce the aging of polymers, which is a major problem sometimes. With time, there will be certain amount of aging due to oxidation and reduction reactions which are taking place, especially if the polymers are present in the highly oxidative environment. So if lignin is present along with these polymers, they can act as antioxidant and reduce the oxidation of the polymer. The lignins have also been reported to improve the properties of various biopolymers such as starch and gelatin, and synthetic polymers such as polyvinyl alcohol, polyethylene, polylactic acid, and polyvinyl chloride. So I had one of the products on polylactic acid and lignin combination for preparation of various different composites, including mulch. Lignin is also used as filler in phenol, formaldehyde resin, and polyurethane. Uh, Urethanes. We will be seeing that also in next slide. Another source use of the lignin as activated carbon. So the lignin can be processed to obtain activated carbon by physical and chemical activation. Excellent absorption properties of activated fibers make them useful in various industrial applications. These activated carbons are used for treatment of sewage and other liquid as well as gaseous effluents. So they can even bind the gases, and those gases of noxious gases, they can be bound by the activated carbon. Then various colored substances or effluent which are present in the effluent which are toxic or are waste. In that case, they can be also bound by the activated carbon and they can be used for purification purpose. So these activated carbons have many different applications, including the decaffeination of various beverages, then metal extraction, finishing of metal for purification of electroplating solutions, for making uh, for making air filters for gas masks and respirators, medicine and pharmaceutical industry, absorption of bacteria, absorption of volatile heavy metals, and combustion products generated by the waste incineration. Okay. Okay. The aromatic structure and numerous functional groups of lignin has attracted increasing attention of its effective utilization in film forming, development of polymers, adhesives, coatings, various additives, and carbon fibers, activated carbon, and as corrosion inhibitor. The addition of lignin enhances the hydrophilicity, biocompatibility, biodegradability, and thermal stability of the composite. Lignin thus find application as plasticizer in composite. Currently, different type of lignins are used for the synthesis of interpenetrating polymer network, which can consist of two or more polymer network. For instance, lignin and xanthangum as superabsorbent hydrogel. Due to its antioxidant property, it is also blended with many polymer matrices like polystyrene, polypropylene, and natural rubber. So, one of the advantage with the lignin is that, as we have already seen, that is, it has got a polymeric structure with multiple phenolic residue components, and with these multiple phenolic components monomers combined together with the carbohydrate. If carbohydrate is removed, in that case, it can have the potential to combine with many different type of molecules with different type of bonding. So these covalent and non-covalent interaction with different molecules help uh, in of as a filmogenic material. It can be useful in polymers. It can be useful as a binder. It can be converted into carbon fiber. So many different applications are there due to its multifunctional groups which are present in its structure. So these are summary of some of the applications like biopolymer formation as a antioxidant, as antimicrobial agent, as plant growth promoter, as metal chelating agent, as a surfactant, and for synthesis of nanofibers, carbon fibers, and tube as aerogel, hydrogel, and as composites. Now, if you transform lignin or break the lignin or modify the lignin using chemical processes or biological processes, they can be converted, for example, by pyrolysis process. They can be converted into acetic acid, phenol, substituted phenol, carbon monoxide, etc. Then, for by fast thermolysis process, they can be converted into acetylene, ethylene, alkali fusion process. They can be converted into phenolic acids, catechol, by enzymatic oxidation processes, oxidized lignin for paints and coating. Then using microbial conversion processes, they can be converted into vanillic acid, ferulic acid, comeric acid, and other acids. So these are some of the hydrogenation and hydrolysis in oxidative processes can also be used for formation of phenol, chrysols, vanillin, dimethyl sulfide, dimethyl sulfonide, sulfoxide, etc. So many different type of products can be obtained by lignin transformation. This shows the lignin product market in terms of revenue share, etc. So carbon fiber is a major market. Then animal feed is there. Then carbon plastics or activated carbon, vanillin. Okay, so many different application and their lignin product market share. We are talking of the economical benefit coming from the lignin. These are some of the companies which are involved in the uses of application and their conversion into different type of products, including Borgard, Dignotan, then Aditya Birla, Wisdom, George, and Ingevity, etc. Now we can also think about a fractionation process which involve lignin and cellulose together, integrated process. It has been reported in which the lignin and cellulose can be fractionated by various processes, including the acid process or alkali extraction process or steam fractionation process. So, in using any of these processes, we can fractionate it, convert into two different fractions. One is lignin-rich fraction, second is cellulose-rich fraction. Cellulose can be converted into glucose or other oxidizable substrate using the saccharification processes, and these can generate the sugar-based building blocks, which can be used for synthesis of many different carbohydrate-like material or many other different uh, polymeric substances. Or the lignin fraction can be treated with the enzyme, and the degradation product can form the precursor of various different value-added chemicals. This is one of the example where enzyme digestion can lead to the formation of fermentation, distillation, and finally the products. One of the example of how the bioethanol and power can be can be created. So raw biomass can be uh, used, uh, and then can be converted into hydrolytic. It can undergo hydrolysis, and after fermentation, they can be converted into and subjected to distillation, and then converted to dehydrated product. And after dehydration, they can be converted into adopted as a fuel, and then that fuel can be used heat and for heat and power production. Thank you. This is another example of the how the lignocellulose waste can be converted. So wood or wood chips which you obtain can be subjected to kraft pulping process, and from which the pulp can be used for paper production or polymer cellulosic fiber production. And this the remaining black liquor will undergo processing uh, through the lignin plant to form the lignin, and this lignin can be either oxidized to obtain the phenolic compound or broken product of biotransformed product of the lignin, or this lignin can be directly used for production of biopolymers. Lignin can also be used for as a source of energy or for production of the vanillin and other products. Okay, this is another example where directly the wood lignin is treated with the ionic liquid and the catalyst to break them into aromatic products. Now, this is how these are the application of the lignin which we are discussing. Now, what exactly we have done is that during my PhD work, we were interested in microbial degradation of lignin, so that the extraction of cellulose from the agro waste becomes easier. So we used various enzymes on the microorganisms so that the lignin can be degraded and removed either during the biopulping process, during the biobleaching process, or in the treatment of kraft effluent. During many of these treatments, our sole aim was to degrade lignin so that, or partially transform lignin so that the lignin carbohydrate complex can be separated. And once the lignin carbohydrate complex is separated, in that case, the extraction of cellulose becomes easy, a lot more easier. It requires less chemical, less harsh condition to remove and extract the cellulose. Later on, we started also working on the hemicelluloses and breakdown of the hemicelluloses. Sometimes because the linkages between hemicellulose and the lignin, so that hemicellulose linkage, like xylanase enzymes, were used for breakdown of the major hemicellulose, that is xylane, and thereby liberating the lignin. Later, we thought that if the lignin can be also removed, why to waste it and start using it? But one of the major issue with the lignin is that when you extract the lignin, it is modified in such a way, depending upon the chemical or physical processes which you are doing, that many of its functional groups are broken down or are converted or modified. So it may or may not act as a useful product. So this that is why we use many different extraction processes for extraction of lignin. And this extraction, after extraction, the different extracted lignin were then used for various different products. So I'll be talking of these products and the applications which we have used one by one. Now, one of the application of work which we have done is that in which the physicochemical property of lignin alginate base film were prepared, and their properties was assessed in presence of different plasticizers. Okay. Now, a lignin alginate blended film was prepared in this work, in the presence of different plasticizers like glycerol, epichlorohydrin, and polyethylene glycol. And the effect of each plasticizer was studied on physicochemical properties of the blended film. Lignin extracted from acacia wood by alkali extraction process was blended with alginate to obtain lignin alginate film in the presence of different plasticizers. A film plasticized with glycerol displayed higher solubility and swelling percentage as compared to EPC and PG plasticized films. The highest tensile strength was also observed for a film plasticized with PG, and none of the plasticizers made any significant change on the bursting strength of the film. So the strength properties were tested after using different type of plasticizer. Now, what are the plasticizers? The plasticizers do not allow the agglomeration or clumping of the monomeric substance when the polymerization is taking place, so that the uniformity of the polymers can be achieved. The incorporation of the lignin considerably improved the light barrier properties of the film, and the Fourier transform infrared spectroscopy study of the film suggests the existence of hydrogen bonding between lignin alginate in the presence of plasticizers. In addition, the epichlorohydrin plasticized film displayed higher uh, thermal stability as confirmed by the thermogravimetric analysis. Further studies demonstrated that the plasticizers significantly affected the physicochemical properties of the blended film. Now, in this work, we concluded that the lignin alginate plasticide with EPC presented better physicochemical and light barrier properties which could be used in packaging and coating applications. So, alginate is a polymeric substance, a carbohydrate. Lignin is a polyphenol, and as the lignin can easily form complex with the carbohydrate in situ condition. So we assume that when we use another carbohydrate and combine it with lignin, obviously the polymer will be formed. Alginate itself can also form a polymeric network, but when you add lignin and other plasticizers, it renders some of the improvement to the property of that particular polymeric film. And by rendering that improvement, like thermal stability, and also the light barrier property, the this biodegradable polymer quality and strength can be improved. This is a different type of polymer and different ratio. We have used this machine. Uh, okay. By using the electro uh, composite synthesis, we have made the nanofibers, and this is an example of a chitosan system. But this can be used for making of the nanofibers and polymers. These are the example of lignin alginate cross linked film with different plasticizers. We have seen the physicochemical properties like thickness, moisture, water solubility, swelling property, bursting index, and tensile strength. One of the reasons of assessing these properties was that when you use a material for packaging material, uh, important thing is that the packaging material should, beside the tensile strength and burst index, that is strength property, they should be able to load, pick up heavy load. Beside that, they should also not absorb a lot of water, and they should not be water soluble. The swelling property should be minimized. The bursting index should be high. The tensile strength should be good. And then only that particular polymer can survive as a packaging material. So we don't want that it should absorb water and get solubilized when you're using it as a packaging material. It should also not allow the light or ultraviolet light to penetrate through and reach the food material, because ultraviolet light also tend to uh, decrease the shelf life of the food material by causing undesirable chemical reaction in them. These are again the atomic force microscopy of the surface of the film. And this is the antioxidant property of this films which we have studied. And this again is the light absorption which we have seen that in presence of excess amount of light reaching the food material, it can be spoiled fast. And this spoilage is not microbial related. It is the physical process by which the ultraviolet, by imparting its energy, can break many of the bonds and thus thereby create reduce the uh, properties of the food material. You can decrease their shelf life. So if you can block this ultraviolet rays from reaching the food material, obviously the shelf life of the food material will increase. ठीक? This polymer can also be used for drug release. The reason being that many of the drugs have to be maintained at a high concentration in body, but what we want is that during digestion and other processes, uh, the drug should not be metabolized, that is broken down, and it should it should be slowly released and maintained in the body in the circulatory system. And when it's maintained at a high concentration at the threshold level for longer duration, then only it remains effective. So we also use this polymer as a carrier of the drug. Okay, so this is how we have done. In the next experiment, where we have used the rice husk extracted lignin. I was talking about rice, that rice can also be used for extraction of many different useful components. For example, the rice extracted lignin and TUS composite. Excuse me. Uh, rice extracted lignin and TUS biocomposite uh, have been used for seeing the effect of acetylation and silane surface treatment for application in the nickel removal. This work was published in Biotechnology Report as a good journal. In this work, a novel lignosilicate composite was synthesized from pre-extracted lignin of rice husk using TUS as a metric. Okay, this is in this we have used rice husk, extracted lignin, and TUS biocomposite. Okay, so uh, and this the extracted lignin was subjected to surface modification by acetylation, followed by in situ synthesis of lignin silicate complex and composite by sol gel method for application in the nickel removal. This lignosilicate were characterized by the FTI. As you remember, that lignin not only forms complex with carbohydrate, it also forms complex with the silica. So silica is also a byproduct of the, especially the agrobase. This lignin silicate was then characterized by the FTIR, scanning electron microscopy, differential scanning calorimetry, particle size distribution, and atomic absorption spectroscope. The FTIR study showed acetyl group in the range of 1680 to 1690, whereas for adsorbed silica nickel, the band appeared at 870 per centimeter. So the findings suggest that the alkali extracted lignin silicate with mean pH of 14.89 nanometer are thermally more stable than ethanol lignosilicate and hot water lignosilicate. So they have higher potential for nickel removal also. SAM and PhD analysis also confirm that they have rough surface and dispersive nature, respectively, suitable for metal bioabsorption. So these are some of the applications about which we are talking about. That is removal of metal, that and then as agent for drug delivery, for antimicrobial activity and plant growth promotion. These are nanoparticles and nanocomposites which can also be formed using the lignin. This is the uh, lignin to use hybrid uh, untreated and treated systems. For nickel removal and the metal removal, we can create the lignin silicate composite and modified lignin silicate composite. Pass and the metal solution can pass through the column, and this element can be then tested for the removal of the lignin. And this complex which is formed will be able to bind to the metal and then remove those metal. This another thought was that the humic acid, which is formed, and fulvic acid, which are component of humus, these are normally present in the uh, mm, soil system, and a part of the humus is formed from carbohydrate, and rest is formed from the lignin and tannins, which are polyphenolic in nature. Another, due to this phenolic structure of the humic acid and fulvic acid, we had an aromatic structure of this humic and fulvic acid. We had the hypothesis because they have similarity in structure, and because these humus material, humic and fulvic acid, have the tendency, have the ability to improve the plant growth. So why not lignin or the modified lignin? So in that case, we have used the modified lignin TUS and LTC, that is the lignin TUS complex, for improvement in the root growth. Okay, root growth. And we also found that in presence of lignin and TUS, especially in the lignin TUS complex, the maximum root growth can be obtained. A TUS in itself can be sometimes toxic, so the growth may not be large. But with lignin, when the TUS is complex, the root growth is amplified. This is a UV visible spectrum of the lignin TUS composites, FTIR of lignin TUS composites, DSC and particle size distribution, then FTIR. Another work which we have done is synthesis and characterization of the cassia lignin gelatin film for its possible application in the food packaging. So earlier we have used the alginate, which is a carbohydrate. Here we have used the gelatin. So this gelatin can also combine together with the lignin. Obviously, the lignin is a multifunctional molecule. So it has the propensity to, under suitable condition, to combine with either protein and it can also be used for precipitation and binding of protein. So that is why we have used cassia with gelatin. And this work, the aim was to develop acacia, lignin, and gelatin blended film using glycerol as plasticizer, and to establish correlation between lignin contents and structure, thermal and mechanical property of the film. Acacia lignin extracted by alkali method was used for the preparation of lignin gelatin blended films by solution casting method. And the result showed that solubility and swelling test of the films concluded that lignin incorporation reduced water affinity of the film, and lignin incorporation produces a noticeable plasticizing effect on the blended film, showing optimum values for the film are incorporated 20 and 30% weight by volume lignin as deduced from their mechanical and thermal property. Lignin blended film had lower glass transition temperature as compared to the controlled gelatin. Infrared spectroscopy of film suggested that lignin interacts with gelatin by hydrogen bonding and hydrophobic interaction, consequently creating conformational changes. Atomic force microscope study displays smooth surface of the synthesized film. Light barrier properties of film revealed that the lignin addition improved barrier properties against UV light in the range of 280 to 350 nanometer. Furthermore, the lowest scavenging activity was observed in lignin and E, that is in which the modification, and tailed by lignin D and C. The lignin gelatin film show improved light barrier and antioxidant property with low cytotoxicity and display great potential in food packaging and coating for preventing ultraviolet induced lipid oxidation and with an extended biomedical application. So this is the work we have done with the gelatin, and then this are some of the work. Okay, this is the TGA, this is the DSC profile of those films, TGA profile, then FTI spectra. Another application of lignin which we have tried is the use of lignin as resin or adhesive. In this work, resin was we have used the alkali lignin which was extracted from acacia here. Uh, so we wanted to prepare the eco-friendly adhesive based on the acacia lignin. So this resin was synthesized using lignin as a replacement of petroleum-based phenol through condensation polymerization reaction catalyzed by characterization was performed using FTI, DSC, TGA by the measure of glass transition temperature, etc. This revealed that acacia lignin resin resembled with control resin along with the higher reactivity. It also exhibited higher glass transition temperature as compared to commercial ligno or sulfonate-based resin. Uh, and the formulated acacia lignin-based resin exhibited uniform morphology and enhanced temperature stability compared with the commercial lignosulfonate-based resin. Further, the highest material absorption capacity was presented by alkali lignin, followed by the lignosulfonate-based resin. The improved properties of the product were attributed to the increased covalent interaction between phenol, formaldehyde, and lignin. Thus, the lignin-based resin can be utilized as adhesive as well as for formation of composite material. In fact, we have tried to do that also in our lab and in experiments. We have tried to use this experiment in our MSc courses, wherein we have used the lignin as a material for synthesis of resin and polymeric material as well as for synthesis of adhesive. And these adhesives are and then used for sticking of the tiles, wall tiles, as well as for wood material. So we have been successful in achieving the proper binding of wood material and as well as the tiles, floor tiles as well as the wall tiles using these resin materials. These are the tentative mechanisms which we have proposed in synthesis of this resin. Then FTR spectrum of acacia lignin control and lignosulfonate based resin, DSC, then thermal degradation or TGA was also done. I already discussed what we have achieved in our result. Another work was synthesis and characterization of crystalline carboxymethylated lignin nanocomposite for metal absorption and antibacterial activity, in which we have used the lignin extracted from rice straw, which was then surface modified using carboxymethylation and then combined with the lignin to form the TUS to form the composite. These composites were then characterized by UV spectroscopy, FTI, SEM, XRD, AS. AS was used for seeing the removal of the metal ion using this composite. The average diameter of this composite was between 160 to 560 nanometer. The XRD and SEM confirm the high degree of crystallinity and porous nature of the nanocomposites with increasing concentration of TUs. The composite exhibited 70 to 82% removal or absorption of the nickel and cadmium. It was also further investigated to assess their future application as wound dressing and antimicrobial and packaging material. Based on antimicrobial and potential to remediate toxic heavy metal, the composites were proposed to be used for wastewater treatment, as packaging material, and as uh, for preparation of biofilters. These are the carboxymethylated lignin which we have used with the increasing concentration of TUS. Okay, these are the visual appearance and visual appearance under the SEM and the antimicrobial activity. As you can see that at a particular concentration they have high antimicrobial activity. These are the FTIS, then SEM showing the surface properties, then XRD and adsorption of nickel and cadmium. The effect of different conditions like shaking time and on absorption of nickel and cadmium. Okay, this is another work which you have already discussed, that is lignin husk and TUS composites, and they are used for removal of lignin and nickel. This is the work in which we have used a nanocomposite with selective antibacterial action and low cytotoxic effect on eukaryotic cells for packaging material and or for wound dressing material. In this, what we have done is that besides the synthesis of this composite, we have tested their antibacterial activity against some of the test organisms like Pseudomonas and E. coli and Bacillus subtilis and Staphylococcus aureus. Uh, uh, and we found that they were showing antibacterial activity. However, they did not show any cytotoxicity against cancer cell lines, including the breast cancer cell lines, lung cancer cell lines, prostate cancer cell lines, etc., and hepatic stellate cells. Thereby suggesting that the nanocomposites can be used as antimicrobial agent for wound dressing with their potent antimicrobial activity, but with low cytotoxicity to the non-target eukaryotic cells. Okay, this is the process by which the composite was prepared, and this is the overall visual observation, how exactly they look like. Okay, this is the electron microscopy structure. Another recent work is green synthesis of silver nanoparticle using acacia lignin, their cytotoxicity, metal sensing ability, and antibacterial activity. This is again an international collaboration which we had for the work. In the present study, the in vitro cytotoxicity, sensing property, catalytic and antibacterial activity of lignin functionalized silver nanoparticle. So the silver nanoparticle are quite common. We can prepare it by any of the process, including reduction or oxidation, using any oxidative or reductive material. But the problem lies with their agglomeration and loss of the property. So if you keep them for a longer duration, the silver nanoparticles tend to lose their property. They agglomerate, that is, form clumps. So to avoid that, you need to modify them. And if you need to add some important property in that, improve their property, you again need to functionalize them. So in this particular work, we have functionalized it using the lignin, and this synthesized silver nanoparticle displays spherical shape and phase center cubic structure with average size of 10 to 50 nanometer. In vitro cytotoxicity assay using MCF7 cancer cell line revealed that the extracted lignin was non-toxic, while AGNPs displays toxicity at both 5 and 50 microgram concentration respectively. Evaluation of the sensing property of the silver nanoparticle that suggested that silver lignin stabilized silver nanoparticle have potential to detect the concentration of heavy metal up to nanomolar range. So these lignin functionalized nanoparticles were used for detection of the uh, of a specific metal ion. How exactly detection takes place? Was when as you know that nanomaterial due to the quantum realm, quantum effect, they uh, their surface and charge ratio, or electron ratio, deviates from a normal uh, mm, macromolecule or micromolecule. And due to that deviation, they have changes in color and texture. And when the binding of a metal ion with this silver nanoparticle takes place, this again changes. This again deviates. So, if there is a change in the color, if there is a change in overall pattern or spectroscopic property of the specific nanoparticle taking place at a given concentration of metal ion, it means the metal ion is interacting with the nanoparticle and causing that particular change. So the sensitivity or the affinity of the nanoparticle or the lignin functionalized nanoparticle for that particular metal ion is quite high. So in that case, we can use this nanoparticle solution as a sensor for detecting the presence or absence of toxic metal ion. It also showed a good catalytic activity with rate constant of 0.00098 and 0.0125 per minute, respectively, for methylene blue dye and paranitrophenol reduction. That is, it was also used for the catalysis of dyes. Okay, so the silver nanoparticle thus displayed antibacterial efficacy against both the gram-positive and negative stain. These findings suggest that lignin-mediated AGNPs can find application in different fields, including biomedical field, drug delivery, textile, biosensor, food packaging, etc. This is another recent work in which the synthesis and characterization of silver nanoparticles loaded with polyvinyl alcohol, lignin electrospun nanofibers, and their antimicrobial activity was seen, in which we have used the lignin and polyvinyl alcohol solution together and the pre-synthesized silver nanoparticle. They were then after interaction at 80 degrees, they were then subjected to electrospinning, and by which the lignin nanofibers, PV nanofibers, were formed. Okay. These were then these nanofibers or the lignin polyvinyl alcohol nanofibers were loaded with the silver nanoparticle, and we then tested them with, for various physicochemical properties. We found that they have the diameter in the range of 100 to 300 nanometer, and the silver nanoparticles were embedded in the structure of the uh, of fiber, and the antimicrobial activity of this fiber containing silver nanoparticle was tested again, Bacillus circulans and E. coli, and they have shown significant result against them. So they can have potential application as membrane filtration, antimicrobial fabrication, wound dressing material. These are the fibers which we have found. Okay, using electrospinning system. Then this is again the structure using this microscope, electron microscopy, individual nanoparticle and their presence. This is again which we have already discussed, synthesis and characterization of crystalline carboxymethylated lignin to use nanocomposite for metal absorption and antibacterial activity. Another property which we have seen that especially the babool, which we have discussed in earlier slides, or acacia, they have many medicinal property. In this, we have tested the free radical scavenging and reducing power of acacia nilotica wood lignin. So lignin is the major component in the acacia, which shows the antioxidant activity. So thus we have extracted those lignin using various methods like alkali, hot water, and organosolvent method. Different fractions were obtained, and then we have tested their phenol and phenolic hydroxyl group content, which normally shows the high antioxidant activity. And then we tested their anti-radical activity or free radical quenching activity. We found they have not only free radical quenching activity, but also have high ferric and molybdate ion reducing capability. The extraction method may also affect the kind of uh, uh, antioxidant activity they will have or free radical quenching activity which they have. In this, we tested the in vitro evaluation of antioxidant and cytotoxic activity of the lignin fraction extracted from the Acacia nilotica. In this, we have also tried the pressurized solvent extraction method and successive solvent extraction method for extraction of the lignin. And then we tested their ability to scavenge nitric oxide, hydroxyl, and superoxide radicals. And we found that they display high ferric ion reducing capability and have excellent potential in the hydrophobic system. Uh, the fractions extracted by polar solvent has highest iron chelating activity as compared to other fractions, indicating ferric ion chelating activity as compared to other fractions, indicating their effect on the redox cycling of the iron. They also showed a higher cytotoxic potential toward breast cancer cell lines, and they were also effective against normal primary human hepatic stellate cells. These findings suggest that the lignin extract of milk would have a remarkable potential to prevent disease caused by overproduction of radicals and also seems to be a promising candidate as natural antioxidant and anti-cancer agent. Then in this work, we have modified the lignin. This was again published in International Journal of Biological Macromolecules. In this, we have used the enzymatic system from Aspergillus flavus and Emershalian illum for modification of the structure. And this structural modification was compared by FTA and C13 solid state NMR. So in this, we have tested after modification and before modification the antioxidant efficiency, reducing power, and hydrogen peroxide scavenging power. And we found we also tested the anti-diabetic in vitro anti-diabetic activity in terms of in vitro glucose movement inhibition and alpha amylase inhibition ability. We found that the structure and functional modification in lignin significantly affected its bioefficacy in terms of antioxidant and antibiotic activity. That is, it improves the activity. This is again a work which was published in Resources in Bioprocessing. That is the synergistic antibacterial and antibiofilm activity of silver nanoparticles biosynthesized by lignin degrading fungus. Okay, in this work, silver nanoparticles were fabricated by enzymatic reduction of silver nitrate using two lignin degrading fungus, that is Aspergillus flavus and Emershalian. The prepared silver nanoparticles were characterized by different spectroscopy, were prepared by characterized by different spectroscopic techniques. Antibacterial activity of prepared silver nanoparticles were demonstrated against E. coli, Pseudomonas aeruginosa, and S. aureus in terms of minimal bactericidal concentration as susceptibility constant. The synergistic antibacterial activity of silver nanoparticle with 4 conventional antibiotics were also determined by fractional inhibitory concentration index using the checkerboard microdilution method. The anti-film biofilm potential of silver nanoparticle was also tested. The FTI confirmed the nature of the capping agent as protein and indicate the role of protein, that is the enzyme, in reduction of silver ion. The average particle size and charge of synthesized silver nanoparticle was 100 nanometer and 20 millivolt respectively. The XRD and TEM also confirm the purity, shape, and size of silver nanoparticles. The EDX data was used for to validate the biological synthesis of silver nanoparticles, and the low MBC and high susceptibility constant indicated high antimicrobial strength of these nanoparticles. So we also found that the silver nanoparticle can also show a synergistic effect with the antibiotic, and the A flavus and emercial and nudulence mediated biosynthesized silver nanoparticle had significant antimicrobial activity and demonstrate synergistic effect in combination with antibiotic. It suggests that nanoparticle can be effectively used in combination with antibiotics to improve the efficacy of antibiotics against pathogenic microbes. The substantial anti-film efficiency of biosynthesized silver nanoparticle can also be helpful against sensitive and multi-drug resistant strains. This is another work which we have recently done. It is on synthesis and characterization of lignin polylactic acid film as active food packaging material. In this work, we describe the synthesis, characterization, and antimicrobial activity of lignin polylactic acid blended film. The film was prepared using co-polymerization reaction of lignin and lactic acid in the presence of stannous chloride as catalyst and polyvinyl alcohol to get a uniform film. The properties of film was studied by uh, SEM, XRD, DSC, DGA. The lignin PLA film demonstrated a significant decrease in swelling ratio and tensile strength in contrast to PLA film. SEM analysis revealed proper blending of lignin into PLA matrix with slightly rough surface. The film was thermostable and amorphous in nature as confirmed from the thermal study and XRD. Further significant improvement was observed in antimicrobial activity and biodegradability of PLA film after packaging after addition of lignin, which suggests the putative application as food packaging as mulch film material. So we wanted it to act as a mulch film also. The reason being that if you use it as a mulch film in the horticulture, as you know that mulch film is required for a specific season so that the weeds are not grown between the plants. So these films normally use are plastic sheets. So if you use this particular mulch film, the biodegradable mulch film, even after the season, if you use suitable degrading material, they can be dissolved and be degraded and act as a fertilizer in the plant system. Okay. So this was one of the main objectives that biodegradable film can be produced, which can act as a mulch film. Other work include the hydrogen peroxide sensing and cytotoxicity activity of acacia lignin stabilized nanoparticle and biodegradation of malachite green by the lignolytic fungus Spigillus flavus, which we have also given a mechanism by which this particular fungus can degrade the lignin. These are some of the lignin-based products which we have made. Raw materials and extracted biomaterial-based resins, foam, films, etc. These are commercially available, some of which form like carbon fiber, lignin carbon fiber-based cars, road dust controller, aerogel. They are extremely light than concrete additives because the lignin have tendency to bind with the calcium and the metal ions, so it can uh, uh, inhibit the fracture formation, so it can reduce the fracture formation. We already seen that they can be converted to carbon black and hydrogel, etc., and disposable materials which can be used. Thank you. So, if you have any queries, I'll be happy to answer them.
The attending members are requested in case if you have any query or any doubts, you can always unmute yourself and talk to our resource person directly. Or you can also type in your query in the chat box, whatever suits you best. So sometimes it takes time for them to type in the query in the chat box.
Yes, yes.
So let's wait for a few
I'll be
seconds.
I'll be here. So,
Well, I guess, sir, that there are no more queries.
no, I think because today is Christmas also. You have
Yeah.
possibly asked them to attend the.
Okay, sir. So on behalf of Ballarpur Institute of Technology and on behalf of the organizing committee of the short-term training program, I thank you very much, sir, for conducting such an in-depth session. Uh, and we are really thankful to you for taking out time out of your busy schedule and conducting such a wonderful session for us, sir. Thank you so much.
Thank you, Bhim. Thank.
Now, to all the attending members, a feedback link will be shared shortly on the WhatsApp group, and you can share the feedback of this session using the WhatsApp link. Please submit your feedback before 11 p.m. tonight, after which the link will cease to accept responses. So you are all requested to please fill in the feedback form before that. The link to share feedback form will be shared shortly on the WhatsApp group. Also, we will now take a short break as per the schedule, and we will start our session after at half past two, that is 2:30 p.m. So you are all requested to join us by quarter after 2 p.m., which is 2:15 p.m., so that we can start the session at 2:30 sharp. So you can use the very same link which has been shared with you now. The link that you have used to join now can be used again to join this session. And it is up to you if you wish to stay in the meeting or if you wish to disconnect and join us back at 2:15 p.m. So in case of any queries, you can always get in touch with our honorable coordinator of the short-term training program, Dr. Archana Nimkar ma'am, or you can also get in touch with our honorable co-coordinator of the short-term training program, Mr. Neeraj Singh Baish sir. Thank you very much for being a part of this session, and see you soon in the next session that starts in 45 minutes from now, that is 2:30 p.m. Thank you so much.